Professor of Immunology at the University of Copenhagen. And it's my pleasure to welcome you to this assessment and defense of a thesis. In a short while, Nina Yen will defend your thesis with the title, The Protective Role of Systemic and Tissue-Resident Th1, Th17 T cells against infection with Chlamydia trachomatis in order to obtain the Ph.D. degree at the Faculty of Health and Medical Sciences. And the supervisors during the Ph.D. study has been the principal supervisor, Jan Prausgaard Christensen from the University of Copenhagen, and the daily supervisor, Jes Dietrich from the State Serum Institute, where the work has been done. And the assessment committee, which I have chaired, means that I've done very little, consists of Martin Tolstrup from the University of Copenhagen and Professor Ali Harandi from the University of Gothenburg. And we will have a defense that lasts at most 3 hours. Of those, 45 minutes allotted to you, that you can present your thesis. You don't have to use all of it, but you're allowed to. And then each of the assessors will spend 30 to 60 minutes discussing the thesis and the presentation. And then the audience can ask questions. And when that is done, we'll conclude the session and the assessment committee will retract and discuss how it went. And then we'll return to you and give you the final recommendation. So with this, Nina, please, the floor is yours.
Thank you. So today I want to present you my thesis that I've done at the Serum Institute with the collaboration of the University of Copenhagen, where I've been enrolled at the Graduate School of Health and Medical Diseases. So my PhD projects have been exploring the importance of local immunity and the role of Th17 T cells against infection with Chlamydia trachomatis. And my overall goals in the PhD project has been to identify an optimal chlamydia vaccine strategy and also to characterize protective CD4 T cells against infection with Chlamydia trachomatis. And with that in mind, I also have the long-term goal, or with the research group, to develop a vaccine to prevent chlamydia-related diseases. So during my PhD, I have written 1 PhD thesis. I have published 2 scientific peer-reviewed papers. And then in the PhD thesis, I've done 1 unpublished studies. So the agenda for today is to introduce you to chlamydia and also what a future vaccine may look like. And then I will go through the main findings and conclusion of my 3 studies, and then conclude on everything and relate to the importance to the field. And then I will go through my future research perspectives. So why is chlamydia a public health issue? So Chlamydia trachomatis is an intracellular bacterium, and globally every year 131 million cases occur. And Chlamydia trachomatis can infect the epithelium of the genitalia, eyes, and rectum, as well as cells of the immune system. And we have different serivars of chlamydia, which causes different diseases. So serovar A to C causes trachoma, which can lead to blindness. And then serovar D to K can lead to this harmful inflammation that we see in the female reproductive tract that can lead to different kinds of diseases such as infertility and ectopic pregnancy. And then you also have serovar L1 to L3 that can cause these lymphogranuloma venereum diseases, and especially occurring a lot in men who have sex with men. So at the moment we have current treatment against chlamydia. However, over half of the cases, people are asymptomatic, meaning that it can be very difficult to diagnose and then treat with the antibiotic. And also, we also see that many individuals get infection because they lack or only have partial natural induced immunity. So there is a global consensus that a vaccine should be developed to prevent infection and disease. So when talking about chlamydia vaccine, we have to focus on different things about the vaccine. So we have to look at what kind of response should we induce. Should it be a humoral response with antibodies along or with cell-mediated response, maybe CD4 and CD8 T cells, and where should we induce this immunity? Should it be locally, or is it sufficient enough with systemic immunity? And how should we induce this different immunity? Can it be parentally, or should it be locally or mucosally? So in my thesis, I have focused on where we should induce the immune response in regards to the vaccine strategy, and then I have focused on CD4 T cells, the protective foam against chlamydia infection. So there are different targets when you look at chlamydia trachomatis and how it affects the host cell. So chlamydia can exist, or its life cycle is a biphasic life cycle, meaning that it exists as elementary bodies extracellularly and then reorganize into this reticulate body intracellularly that can replicate inside the host cell. So it enters by endocytosis and come in and replicate inside the cell and then reorganize to prepare for exit by lysis or exclusion. So when looking at this, there are different targets you can have with a vaccine. You can target with a vaccine. So the first thing that I focused on is CD4 T cells, and they can secrete interferon gamma, which can activate interferon gamma restrictive mechanisms inside the cell that can inhibit chlamydia growth. And then you can also recruit neutrophils and macrophages. CD4 T cells can do that, and then phagocytize the bacteria. And then also, I also want to mention, we also have the humoral response antibodies, which can neutralize or opsonize the bacteria, or activate or attract the complement system to kill the bacteria. And then you also have the CD8 T cells, which can recognize the infected cells and induce apoptosis. But here I want to focus more on the CD4 T cell side. So right now we don't have any chlamydia vaccine available, even though whether we have different targets with the vaccine. So what do we really know about the protective immunity? So the protective immunity is very established for Th1 T cells. So Th1 T cells are these CD4 T cells which express TFETs and then can secrete different cytokines, and its major effector cytokine is interferon gamma. Interferon gamma secreting Th1 T cells have been shown to mediate growth protection in chlamydia trachomatis infected humans and also in animals. However, recently a study showed that inhibition of Th1 differentiation in a Tibet knockout mice did not abolish protection against infection with chlamydia, meaning that there may be other components of the immune response that maybe won't be protective against chlamydia. So what could that be? So it could be these Th17 T cells, but their role is not so very clear. Th17 T cells are defined by Guapana T expression, which increase expression of the different cytokines, and IL-17 is its major effector cytokine. So Th17 T cells and IL-17 signaling effect on the immunopathology has been studied a lot in chlamydia. Some studies have seen that they mediate immunopathology, while one study saw that there was no association to immunopathology. When you look at the Th1 response, studies have also found that the lack of IL-17 signaling actually decreased the Th1 response inside the genital tract. And there the studies have talked about if IL-17 is important for Th1 recruitment in general, or if it's because the Th17 T cells can transdifferentiate into Th1 T cells. So there's maybe a hypothesis there. Then IL-17 signaling has been shown to mediate protection in other genital tract infections such as herpes simplex virus and Sierra gonorrhea. So Th17 T cells could play a role in inducing protective immunity against genital tract, and maybe a component that we should induce with a vaccine. So how should we induce this? So we have this vaccine, CTH522 vaccine antigen, adjuvant and formulated with type 1 adjuvant to make this vaccine. And the special thing about our CAF1 adjuvant is it's this cationic platform. And the CAF platform were developed to induce these strong cell-mediated mediated responses. And it consists of the surfactant DBA that form the liposome, and in itself it can induce Th1 and Th2. But if you incorporate different immunostimulators, you can further direct and amplify the immune responses. So in my thesis, I've used Kappa1, so I want to go further into that. So Kappa1 consists of DBA together with TDB, which is this synthetic growth factor analog to the mycobacterial glycolipid TDM. And both TDB and TDM have been shown to induce Th1 and Th17 responses by activating the C-type lectin mingle receptor on antigen-presenting cells, which leads to beta activation and transcription of different pro-inflammatory cytokines. And other than that, it also stabilizes the liposome, which leads to less aggregation of the vaccine. So when formulated together with our CTH522, it's a recombinant subunit antigen, which consists of the major outer membrane protein of MUMP, which have multiple T and B cells inside. And then it's decided to also make this recombinant subunit antigen and extended the MUMP along with the variable domains of the regions of the most prevalent serums, which have been shown to induce a lot of neutralizing antibodies. So together, the CTH522 and Kappa1 vaccine can induce Th1 and Th17 responses as well as antibodies. So that leaves us with what vaccine strategy should we choose for a chlamydia vaccine. So there's a lot of studies looking at different vaccine strategies. So a study was reported that intranasal immunization induced both protective local and systemic immunity against chlamydia trachomatis. However, there are safety concerns regarding having such a potent vaccine in close proximity to the brain. Then you also have the intrauterine immunization, which is purely to study the role of local immunity against chlamydia trachomatis. And then at last you have a parental immunization, which is the one that's considered the most safe. However, it was considered up until recently sufficient enough. However, a recent study showed that parental immunization do not lead to induction of local immunity and then do not lead to protection against chlamydia trachomatis. But we maybe think that with our adjuvant and with our antigen, we can try to see if a parental immunization is good enough to protect against chlamydia trachomatis. So how should we evaluate the vaccine strategies? So we have different models available. So we have a vaginal infection model. However, the human strain is quickly eradicated in this model. So if we want to study infection with the human strain, there's a newer model that was developed where you put the infection right in the base of the human horns, and then you can see that effective bacterial colonization is established, and you also see establishment of clinical relevant pathology and infertility. And also it leads to a strong T cell recruitment into the genital tract, where you can study the protective role of those immune responses. So that leads to the project aims. So the project aim was to optimize the transcervical infection model to study vaccine-induced protection, and then examine if a parental vaccine is sufficient for protection, and also characterizing protective CD4 T cells with the focus of characterizing the role of TS17 T cell. So that I did in the first paper, or one of the objectives was in the first paper. So the rationale for this study was based on the recent study that did not show any protection with a parental immunization because of the lack of local immunity prior to infection. But we thought with other adjuvant and antigen, can we then induce systemic CD4 T cells that are able to locate to the genital tract upon infection and then protect. So the objective was to evaluate the protective capacity and also optimizing the transcervical infection model. So the experiment setup is these black 6 FM1 mice that received 3 vaccination and then were infected 6 weeks after vaccination, and then post-infection collected swabs and harvested the organs. And from the organs, we made this single cell suspension and in vitro stimulated with CTH5P2, which were the vaccine antigen, and to look at the vaccine-specific CD4 T cells by measuring the secretion of IL-17, interferon gamma, TNF alpha, and or IL-2. So the vaccine-specific CD4 T cells were the cytokine-positive CD4 T cells we defined as the measurement of the 4 different cytokines, and Th1 and Th17 T cells were divided upon either expressing IL-17 or not expressing IL-17. So the first finding we saw is when we gave an infection with a high infectious dose, 10 to the 5th IFF, we recruited to the genital tract, we see that at early infection at around day 3, we see that they have quite similar distribution as we observed before immunization systemically. If we look at peak infection around day 7, we see that there's an increase now in the cytokine positive, the percentage of cytokine positive T cells, especially the Th1-like and the Th1 T cells. And in general, I showed in the supplementary that all of them actually increase in numbers. But however, the cytokine negative population now makes up smaller numbers of the vaccine-induced CD4 T cells. If you look after clearance at day 22, you see that the picture now goes kind of back to what we saw prior to infection out in the systemic, where we have the majority of the cells not expressing any of the cytokines that we measured. And when we look at even further from that, at day 57, you see that the major population is still this cytokine negative Th17 T cell population. So we wondered what this population was, or if it had any function or it could respond. So we wanted to look at the activation status out in the genital tract at day 57. So here we infected the mice and we waited until day 57, and then in the meantime we gave BRDU before the day 7 organ harvest, and also BRDU before the late time point out here to assess the proliferation capacity at the peak infection and long after infection. So BRDU is this thymidine analog that can be incorporated when the cells are proliferating. Then I also looked at KLRG1, which is this senescent marker that can be upregulated on terminal differentiated Th1 T cells, and it's also this checkpoint inhibitor. And then we also looked at CD127, which is one component of the IL-17 receptor, or IL-7 receptor, which increased T cell survival by upregulating the expression of anti-apoptotic genes, and is also maybe not be targeted for inhibition or T cell senescence. If you look at this 127 expression, we see that it is quite increased from day 7 and day 57 in the cytokine negative population, meaning that maybe you can use that receptor for survival augmentation. Then we looked at BRDU, and you can see that at day 7 it has a lot of BRDU incorporated. Here we also have a lot of inflammation going on and a very quite big response in the genital tract. But at day 7 we still see a BRDU incorporation out in the cytokine negative TS17 T cells. So that could either be from what we saw at day 7 here, or it could be from what we've seen that maybe there's a homeostatic proliferation out in the genital tract. So it could either be that they survive until day 57 or that they are incorporated BRDU. So it looks like that the cytokine negative TS17 T cells they're activated. So we wanted to see how they responded towards a reinfection, and also to see if maybe they could give rise to one of the other cytokine positive TS17 T cells. So when comparing the mice that have been reinfected, the whites, with the red that have not been reinfected, we see a difference not the distribution, but we see a lower level after reinfection, and we see an increase of the Th1 ligand and Th1 T cells, and also, So we can see that they're able to respond still to the reinfection, and it could maybe that these two populations come from the cytokine negative Th17 T cells, but we can't conclude that from our studies. We need to look into that further. So what I can conclude is that the vaccine induces a heterogeneous Th17 population, and the majority of the vaccine-induced CD4 T cells are of Th17 T cell origin, consisting of the cytokine negative and the Th1-like Th17 T cells. And all of the Th1 T cells are able to recruit it and respond, and they also seem to persist in the genital tracts. So to conclude on all of the studies related to the importance of the chlamydia field, we have seen in our studies that a preventive vaccine strategy would be sufficient strategy. We see that resident Th1 and Th17 T cells protects against infection. We see that a parental vaccine can actually lead to this local immunity post-infection. And we also see that the Th17 T cell population consists of several Th17 subsets. So what we want to do further is we really want to see how important the Th17 T cells are for protection, as they constitute such a major population of vaccine-induced response. So we had different approaches. So we want to characterize them by single-cell RNA sequencing, looking at their transcript expression at a single-cell level, enable us to go look into further of this population that we do not know anything about, that they have produced IL-17 at one point. Then we also want to look at the protective capacity by that we transfer the Th17 T cells. And then we also want to look into protection in these T-bed knockout mice when we vaccinate it to see what can the Th17 T cells do without Th1. And then we also want to neutralize the IL-17 or inhibit the IL-17 signaling, and then also induce depletion of all cells with a history of IL-17 expression with this Ph.D. toxin receptor mice. And we already started one of the approaches. So we already tried to vaccinate a T-bed knockout mice. And we saw that if we give them a vaccination, we see that without Th1 T cells there, we can still observe a protection, a significant protection. And if you look at the mice here, you see that they do not express any interferon gamma, which was surprising as it is seen before that they had a huge protective role. If we looked into this population of IL-17 secreting CD4 T cells, we saw that a lot of them expressed also TNF alpha and IL-2 along with TNF alpha. We also did some RNA-based sequencing of the YFP positive CD4 T cells, looking at them in the systemic and in the nodal tract. And we saw that they have an upregulation in several genes from the blood, and when they come into the general tract, they can actually respond by upregulating different genes for immune effector functions and activation and also adhesion. So it seems like the yellow fluorescent protein CD4 T cells, Th17 T cells, they actually do respond as we've seen for the cytokine analysis. Yes, so at last, I really want to say thank you to my supervisors, Jess and Jan, and also our chlamydia group, Shamla and Sophia. And then I thank you to my assessment committee, and also thank you to my colleagues and my friends.
Thank you very much for a fantastic presentation. It was spectacular. And my overall comment when I read this thesis was that there's a lot of hard work in here. There's a lot of experiments. And I know how difficult all these mice experiments. It looks easy when you present it like this, but there's a lot of tips and tricks and long days. And it's an impressive exploration of these CD4 T cells, and it's by no means easy. And I had some problems in understanding it, so that's where my questions are going. So hopefully you'll get it. And there are many techniques that you've shown that you can master, and specifically flow cytometry. And it's a wonderful technique. It gives you a lot of answers, but also quite a bit to take care of when you analyze your data. I'll have some general questions for the background and for the overall topic, and then I will go into a little bit more details about the different papers. But overall, very nice. Thank you. Is it possible? Do you have your thesis present or figures or?
Yes, I have them. Yeah.
I would love to see the figure 4.2, the adaptive immune system. Oh, you probably know it by heart. We can discuss. I have it here.
Okay, good.
So if you can just this you discuss a lot about the plasticity of the T cells.
Yeah.
I mean, this differentiation, and we always like to think of things in boxes. And your figure 4.2, there are some nice boxes for Th1, Th2, etc.
Yes.
Can you just briefly describe the differences as you see it from Th17 cells that you have studied so intensively to the Th1 and the Th2?
So the difference between this Th17 and Th1 and Th2. Yeah. So the first difference is, of course, how they are induced. So with Th17, we induce them by IL-6, TGF-beta, and IL-23. And with Th1, for example, we induce them with IL-12 and actually again, there's also interferon gamma. And then with Th17, we induce the STAT3, and that also helps with inducing gamma T expression that leads to expression of IL-17 and IL-22, and also a little bit of IL-21, and then also GM-CSF which is this granulite monocyte colony factor that activates the neutrophils and recruits them into what is supposed to be included. And then for the Th1 cells, you can use IL-2 and TNF alpha, and also, of course, interferon gamma. And you see that is the traditional roles of the two. Also, Th2, I can't remember what exactly induces Th2, but it can secrete cytokines such as IL-4, IL-5, IL-13, and have also increased responsibility to the antibodies. So with Th17 T cells, they can transdifferentiate into Th1, get a Th1-like phenotype, and start to, as we've seen in studies, that they start to secrete IL-2 and TNF alpha and interferon gamma also. So they can try to go into the different CD4 T cell subsets.
Perfect. You described that very nice. Also, you devote a lot of time in your thesis on this. I think it's very important to see T cells not as defined in boxes, because and then they're sort of stuck there and you think of them as Th1, but what does that mean?
Yeah.
And then you discuss this interferon gamma, so as sort of the primary effector cytokine that you induce.
Yeah.
So CD4 T cells, can they induce cytolysis of an infected cell?
So some studies have seen that they can induce cytolysis, or I don't remember seeing that they induce as such, but I've seen in different assays, such as in vitro killing assays, that if you isolate the CD4 T cells and then in the presence of the target cells, you can see that they can kill their target cells. And a lot of people have also seen, especially have seen that they increase expression of different cytotoxic markers. And there was one paper who described it for Th17 T cells that have an increased expression of this NK2 GRD marker, which is typically a marker that you find on NK cells. So they can recognize this MHC1 B gene that is upregulated on fibroblasts and also epithelial cells. So it could be a way, if the Th17 T cells increase that marker, that it can target these cells that express these MHC1B class B genes, and then induce cytotoxicity. But we also have the MHC2 or the TCR on the CD4 T cells, and CD4 molecule recognize the MHC2. So the target there could be infected immune cells, where you see that especially macrophages, candidate recruiters can reside inside the macrophages and hide in there by inhibiting the fusion with the factosome when they get into the cells. So there are different targets for the CD4 T cells to actually exhibit these cytolytic functions.
So just so you think that some of this, so the protective role in the genital tract could be both release of interferon gamma that would affect the epithelium and other cells, but also a direct cytolytic effect potential.
Yes, potentially, yeah.
So I study some different pathogens, and always when you want to, for instance, develop a vaccine, you really have to know what type of immunity you want to induce.
Yes.
So, and what that leads to is a finding of what is termed of a correlate of protection. What type of immunity that we can measure, that if you have sufficient quantity of such an immunity, it can then lead to protection. Is there a well-established correlative protection in chlamydia so that you know, okay, I have 20,000 units of an antibody that will lead to protection, or I have 20,000 CD4 T cells that will lead to protection? Is such a correlative protection known in chlamydia?
Not to my knowledge, we do not have any correlates of protection that we can say, for example, if you can measure the blood and say this is good enough to protect. So we don't know how much antibodies should be induced, or how many Th1 T cells should be induced, or other kind of immune cells. So we don't know. We don't know that.
And you just strike something, so you say measure in the blood. And you've done a lot of work here to avoid measure things in the blood, which I would like to,
Yeah.
to acknowledge. I mean, it's important to look at the site where the infection occurs.
Yeah.
So is there evidence that what you measure in blood, which is easily accessible, I mean, to get in a mouse is easier than in a human, but then compared to the genital tract for immunity-wise in terms of protection?
In the blood, so there's a lot of human studies that look at especially antibodies and look at the CD4 T cells, but I don't think that people have yet conclusively said if we can measure this, it means protection. There's a lot of human studies where they looked at antibodies against different chlamydia antigens, and seeing that if women express, if women that have been infected express these antigen against, or these antibodies against these antigens, there may be some correlation to protection or a better outcome, against a reinfection, but it's not, I wouldn't say that it's something that is totally conclusive. They have seen that, I think it's antibodies against the heat shock protein 60 or something like that, where they see that if you have an increased antibody response, you may have some partially immunity against, but it's not something that is, I would say, is quite established.
But that would be important to know.
It would be important, yeah.
So that you know what type of, whether you should vaccinate with the heat shock protein or mump as you've done.
Yeah.
So if that is not completely clear, how could you use your model to investigate which immune parameter is most important? What type of threshold that you would do? Could you think of such a study, or have you already?
Yes, I think it's something that at society also, there's some projects going on to do exactly that, where you look after immunization, and then by doing bioinformatics you can see which kind of genes that are upregulated, and then see what the outcome is, and then correlate with gene expression to what the outcome could be, protection or not protection. So trying to correlate what we see in the blood with protection or not protection. So it's something that is definitely really, really important for vaccine developers to have a correlate of protection, and especially just in the blood. So, not that I'm working myself, but we could definitely also use the marine model to maybe have some kind of direction of where you should look at, and what kind of, if we're looking at protective CD4 T cells, maybe look at what kind of systemic CD4 T cells will become tissue resident or induce local immunity.
And I think that discussion, just to wrap it up, so protection can be two things, right?
Yeah.
It can be that you
have sterilizing immunity, or protection against just control of infection and protection against the pathology.
Yes.
Yeah.
And it looks like with your first study that you're actually with the right model
Yeah.
settings,
Yes.
that you can actually induce or avert pathology, right?
Mm,
So that would be protection from disease.
disease, yes.
Do you think that you can achieve protection from infection?
I think it's difficult for chlamydia because when you have sterilizing immunity, you risk the development of pathology. So you want something that is strong enough to decrease the bacterial levels, but you don't want it too strong that it can use immune pathology. So you want something in the middle. So it's not a goal to not have any infection at all. The goal is to prevent the disease. So that is the focus of the chlamydia vaccine. But of course, if you can prevent dissemination to other partners or so on, that would also be really good. But the end goal is pathology, or the pathology development inhibition.
So maybe this is the time to just, so what you, in terms of your model, so you have used this infection model where, there are two things that I would like to ask you. So first, what do you think, so you deliver chlamydia, you bypass the vagina.
Yes.
And what do you think that, how does that mimic the natural infection route in terms of comparing to, what, my question is, what type of immunity, is a difference in a natural infection versus your model in terms of immunity protection?
Yes, it is because we bypass the normal route, like the natural route of infection. So what we mimic is when the infection ascends up to the uterus or the upper genital tracts. So that is what we mimic with the transcervical infection. But it is, if you want to look at this model and compare it to how humans are infected, it is not a natural way of doing it. Because in the vagina you have the barrier, you have the microbiota that is also affecting in infection. It's supposed to protect against infection too. And then you also, of course, also have the mechanistic that needs to travel up to the uterus and then ascend. And we have a lot of immunity there, both antibodies and cell-mediated immune responses. So the model is really good at looking at upper genital tract responses, but in terms of what we call it, to model a real infection, it lacks. Yeah.
And I think that's very important. I mean, a model is a model and it can be very useful. But
Yes.
then, going back to my question before of achieving sterilizing immunity, is probably actually asking a lot for a model where you deliver it. That's that just. Now you presented it beautifully here, CAF01 adjuvant, and I was missing that a little bit in the thesis, but now you made up for it, that was good. So why do you, so it looks like there is a whole lot of different adjuvants to pick and choose from.
Yes.
So why do you think CAF01 is a good adjuvant? What does it do specifically?
Yes. So for chlamydia in general, it reduces, as I said, it reduces both Th1 that people have seen before, play a big protective role against chlamydia. Then it reduces antibodies that have also been shown to have protective role against chlamydia, maybe with some help from CD4 T cells. And then we also have Th17 T cells, which we think have protective role against, could have a protective role against chlamydia, with them inducing interferon gamma and also TNF alpha and IL-2, and a little bit of IL-17. Okay.
We'll not say that. Can we go to your first paper? Because
Yes.
if you pull up the first figure, or the second figure, so I would like to discuss this
Yin. We have discussed and deliberated, and we've decided to recommend that you be awarded the Ph.D. degree. Congratulations.
Thank you. Speech. I can do very short because.
Được rồi.
But I just want to say thank you for being my supervisor and always encouraging me and giving me my confidence. You definitely done that. And, to all of you guys, also thank you. You're the best colleagues ever. And my family also for supporting me and giving me the opportunity to do a PhD and everything. Yes, thank you. Thank you.